Welcome back to Chapter 4. In this chapter, we'll discuss genetic principles in blood banking. These are your objectives. We'll be discussing dosage effect, we'll talk about codominant inheritance, and we'll talk about phenotype frequency and how it's used to find compatible units of red blood cells. We'll discuss the Hardy-Weinberg principle and talk about Mendelian laws of independent assortment and genetics, and we'll define the terms linkage and crossover. A bit about some of the molecular testing techniques that we use for red cell typing and for HLA typing. Blood group systems are groups of antigens on the red cell membrane that share related serologic properties and genetic patterns of inheritance. An example of a blood group system that we've talked about already is the ABO blood group system. The genetic material for our blood group systems are found in DNA. DNA is contained in chromosomes that are in the nucleus of every cell in the body, with the exception of red cells and platelets. Genetic material is replicated by either mitosis for the somatic cells or meiosis for the gametes. Remember that the somatic cells have 46 chromosomes. The gametes only have 23 chromosomes because one gamete will join with another gamete to form a full cell. Now let's talk a bit about phenotype versus genotype. Phenotype is the physical representation of a gene, while the genotype is the actual genetic makeup, or the series of A's, C's, T's, and G's. The physical observed expression of traits is the phenotype. A patient's phenotype is described or determined by the hemagglutination of the red blood cell antigens using antisera. An example of this, a person whose red blood cells are non-reactive with anti-A or anti-B antisera is group O. The genotype, on the other hand, again, is the actual genetic makeup. This can only be determined by molecular techniques or family studies. An example, and this is actually an example of me, a person with the phenotype A could have the genotype AA or AO. Molecular testing can confirm which is present. The difference between genotype and phenotype is illustrated in this diagram of ABO system inheritance patterns. In the first example, we have parents with the phenotype of A and B. Their genotypes are AO and BO. The possible genotypes of the children that they could have are AB, AO, BO, and OO. This unique combination of genotypes can produce children with 4 different phenotypes. In the second example, we see a parent with the phenotype AB and a parent with the phenotype O. The only phenotype for a genotype of AB is the phenotype of AB, and the only genotype of a person who has OO is O. So, with the combination of one parent having AB and the other one having OO, the only 2 phenotypic combinations are A and B, where all of the children would inherit an O from the type O parent. Half of the children would inherit the A from the AB parent, and the other half would inherit the B. So statistically, 50% of the children would be type A, and 50% of the children would be type B. This is another way of looking at the information that we just saw on the previous slide. A Punnett square is used to predict the probability of an offspring's genotype. It summarizes every possible combination of maternal and paternal alleles of a particular gene. In this case, we have the A, B, and O genes. Now let's talk a little bit more specifically about genes. Genes are basic units of inheritance on a chromosome. A locus is the site at which a gene is located on a chromosome. Alleles, which are alternative forms of a gene, are found at each locus. They are codominant. So this example of the ABO gene locus, we have the genes A and B located at the same place. Think of this as alternative versions of a story. You have one story, something happens in story A on one chromosome, and something else happens in story B on the other chromosome. Both of those stories are located at the same place on the chromosome. So if you think of this as a book, both of those stories are located on the same page of the book. They just say different things or they give different instructions. In the case of A and B, both A and B are codominant, meaning that A is going to be expressed equally as B. These antigens produced by opposite alleles are antithetical. So another example is JKA and JKB. Those are located at the same spot on that particular chromosome. Multiple alleles at a single locus are considered to be polymorphic. A recessive gene is a gene that's expressed only when it's inherited by both parents. A codominant gene, which is the example for our blood group antigens, show equal expression of two different alleles. Blood group antigens are considered to be codominant. A dominant gene is a gene that's expressed over another gene. Genes that don't express a detectable product at all are considered amorphic, and the O gene is an example of an amorphic gene. So for our A, B, and O blood types, A produces a gene product, B produces a gene product, but O doesn't produce a detectable gene product. So if a patient inherits A from one parent and O from another parent, the A is the only one that's going to be expressed because there is no actual antigenic product for the O gene. Mendelian principles can be applied to blood group antigen inheritance. Independent segregation occurs when one gene from each parent is passed to the offspring. Independent assortment is demonstrated when blood group antigens from different chromosomes are expressed separately, resulting in a mixture of genetic material. The two exceptions to this law are linkage and crossing over, which we'll discuss in the next couple of slides. First, let's talk about linkage. So linkage occurs when two genes that are very close to each other are inherited together. Genes that are inherited together or linked is called a haplotype. An example of this is the Y chromosome. So the entire Y chromosome is inherited exactly the same by a son as it appeared in the father. So you can trace the Y chromosome because it's all inherited together. There's no recombination with another Y chromosome because each man only has one Y chromosome. So you can see that the Y chromosome of a son is exactly the same as his father, which is exactly the same as the grandfather and the great-grandfather, etc. Some haplotypes tend to occur at a higher frequency than for unlinked genes, and this is a phenomenon called linkage disequilibrium. The example here is the M in big S and little s group. So these are inherited together as a haplotype, and they occur at different frequencies. The frequencies that we see for these linked genes does not follow Mendelian genetics or Mendelian principles, which is our clue that they are inherited as haplotypes. There's more information about this in your textbook. Crossing over is another phenomenon that does not follow Mendelian genetics. Crossing over occurs when two genes on the same chromosome combine and produce two new chromosomes. The example here is that you have an allele on one chromosome labeled A and an allele on another chromosome labeled B. When crossing over occurs, the genes switch the chromosomes that they're on. So at the end of this process, A and B are located on the same chromosome, while the other chromosome has neither A nor B. Most blood group system genes are on autosomes, except for those of the XG and KX systems. Autosomes are all chromosomes other than sex chromosomes. So all of our blood group system antigens are on chromosomes other than the sex chromosomes, with the exception of XG and KX. The XG genes are found on the X chromosome. If the father carries the XG allele, he will pass it to all of his daughters, but not to any of his sons because he doesn't pass an X chromosome onto any of his sons. He passes the Y chromosome to the sons. If the mother carries the XG allele and not the father, all of the children will express XG. We talk a lot about heterozygosity and homozygosity in blood bank because of the thing that we're going to talk about on the next slide called dosage. So a person who inherits identical alleles is called homozygous. This would be if you were blood group type A and you inherited an A from your mother and an A from your father, you would be homozygous for A. Same thing for B. If you inherited a B from your mother and a B from your father, then you would be homozygous for blood type B. A person who inherits different alleles is called heterozygous. So a blood group A person who has phenotype blood group A could inherit the genotype of A and O, meaning they get A from one parent and O from the other, meaning that they are heterozygous for blood group blood group A. This is the same phenomenon for all of our different alleles of the blood group systems. One of the reasons homozygosity and heterozygosity is so important in blood bank is because some blood group systems show dosage. That means that persons homozygous for an allele have a double dose of an antigen on their red blood cells compared with those who are heterozygous for an allele. So for an example, if a patient has a double dose or is homozygous for big C, which is part of the Rh system, then they will have double the amount, roughly, of big C antigen on their red blood cells than someone who is big C little c. Dosage is a variation in antigenic expression due to the number of antigen sites present on the red blood cells. The reason that we talk about dosing is because double dose expression of some antigens may show stronger agglutination compared with antigens that are a single dose. Antibodies to antigens in the Rh, Duffy, MNS, and KID blood group systems demonstrate dosage. This is important when we're trying to identify an unexpected antibody in a screen. This is an example of how we use dosage when looking at an antibody screen. The antibody screen, if you'll remember, is mixing cells with our patient plasma. So in this case, we have one cell that's mixed with our patient plasma, and we get a result of 3+ agglutination. In cell number 2, we've mixed cell number 2 with our patient, and we get a reaction of 1+. This 3+ and 1+ are significant because the reaction is stronger in one cell than another. So what we're looking for, because we suspect dosage in this case, is we are looking for a homozygous antigen in the first cell that is showing a 3+ reaction, and we suspect that there is a heterozygous situation for the second cell. So if we talk about the antigens that show dosage, we have our Rh system, we have M, N, and S, we have Duffy, and we have KID. So if we look here at the M and N, we see a homozygous M with no N, and we see a stronger reaction. This is demonstrating a double dose of M, which is giving us a stronger reaction. This patient also is having a reaction to this heterozygous M, but because the heterozygous state has less M antigen, the reaction is going to be weaker. So homozygous expression of some antigens will show stronger agglutination compared with antigens that are heterozygous. Now, a good example of why we do an antibody panel after an antibody screen, because if you look at JKA and JKB, we have the exact same representation as we do over here with M and N. And so it is possible that the patient has an antibody to M, but it's also possible that the patient has an antibody to JKA. And so we would do our antibody panel to rule in or rule out M or JKA as the unexpected antibody. The location of inherited genes in cis or trans positions can affect the expression of the antigen. Alleles that are on the same chromosome are cis to one another. Alleles that are on opposite chromosomes are in the trans position. So in the first example, we see that we have two genes that are on different chromosomes. Those are in the trans position. Genes that are located on the same chromosome. So we see in the second example of D and CE located on the same chromosome, those are in the cis position. To determine genotype or phenotype occurrence, we use two different formulas in the blood bank. The first one is called the phenotype calculation, and the second one is called the Hardy-Weinberg formula. We'll go through both of these. A phenotype calculation enables finding a unit of red blood cells with certain antigen characteristics like antigen negative. The Hardy-Weinberg formula creates a determination of the gene frequencies that produce a particular trait. Whenever we have a patient with an unexpected antibody to a blood group antigen, we want to make sure that we give that patient a unit of blood that does not contain the antigen for which they have an antibody. The first step is to look up the percentage of antigen or the frequency of that particular antigen in the population. Now, this is going to be different based on where you are across the globe. So if you were in China, for example, you may have much more big S than you did in the US. So this is all completely dependent on where you are across the globe. So you can look up the percentage of positive units of blood or the antigen frequency in a given population. So if you have, in this example, a patient that has multiple antibodies, which this does happen, so this particular patient has anti-big C, anti-big E, and anti-S, and this patient needs a unit of blood. So what is known? We know that 70% of the population is big C positive. We know that 30% of the population is big E positive, and we know that 55% of the population is S positive. What we want to know here, what we're looking for, are antigen negative units. And so we subtract 70 from 100. Those are zeros there. So 100 minus 70 gives us 30%. 100 minus 30 gives us 70%, and 100 minus 55 will give us 45%. We convert that to a decimal and we multiply them together. So 0.3 times 0.7 times 0.45 will give us 0.0945. We'll go ahead and round up. And so our units or our number of percentage of negative units is going to be 10%. That tells us that about 10% of the population will be negative for all three of these antigens, or about 1 in 10 units will be compatible. This means that if we need 1 unit of blood, we will likely have to antigen type 10 units before we find 1 unit of antigen negative blood for all three of these antigens. This is another example of a patient with multiple antibodies that needs 2 units. So we apply the same principles here. 100 minus 66 is going to give us 34. 100 minus 72 is going to give us a negative percentage of 28, and 100 minus 9 is going to give us 91. We convert those to decimals and we multiply them together. So 0.34 times 0.28 times 0.91, those are the percentage of negative units in the general population, is going to give us 0.087 or 9% negative. That means that 9 out of 100 units are going to be statistically negative for all three of these antigens. However, in this case, we need 2 units. So you would divide 2 by 0.09, which is the antigen negative frequency, and that gives you 22. That tells you that you probably need to pull segments or pigtails from 22 different units in order to find 2 compatible units for your patient. And finally, this is one more example where you have 3 antibodies, multiple antibodies, anti-big C, anti-big E, and anti-S. You determine the frequency of the antigen negative units. So you would find the positive frequencies and subtract those from 100. You multiply them together. In this case, this calculation uses slightly different numbers and it comes up with 11%. Still about 1 in 10 units will be compatible. In this case, we need 4 units, and so we take 4, we divide that by our negative percentage here, which is 0.11, and we get 36.4 units. Now, we cannot antigen type 0.4 of a unit, and so we're going to always go ahead and round up no matter what the percentage is or the decimal is there. We're going to always round up. So 36.4, you cannot type 0.4 of a unit or half of a unit, and so we're going to just round up to 37. Now, the Hardy-Weinberg formula is used for Mendelian inheritance patterns, where you have 2 different alleles. P represents allele number 1, and Q represents allele number 2. The gene frequency is expressed by the equation P plus Q equals 1. The genotype proportions are listed here. So P plus Q squared equals 1. You can just rearrange your equation by using basic algebra here, where P squared plus 2PQ plus Q squared equals 1. So the example here, P is the frequency of allele A, Q is the frequency of allele small a. And so if you're given one of these frequencies, you can figure out the other one by plugging into your equation. So what is the frequency of Q if P is 0.3? 1 minus 0.3 is 0.7. And then you can use your basic algebra to figure out the homozygous and heterozygous percentages for those as well. Now let's talk very briefly about PCR, only because we use PCR technology and molecular techniques in our molecular lab when trying to determine RBC antigens. We also use this for HLA typing. So polymerase chain reaction, or PCR, is a method for rapidly and precisely multiplying specific DNA sequences. So with each PCR cycle, we double the amount of DNA. That means that if you initially start out with 1 copy of DNA, after 1 cycle of PCR, you'll have 2 copies of DNA. After another cycle of PCR, you'll have 4. After another cycle of PCR, you'll have 8, etc. So there are 3 steps in PCR. In the first step, your double-stranded DNA is separated into 2 different strands. You then have the annealing step, and the annealing step is where a primer attaches to the end of one of those strands of DNA. So you have a forward primer that attaches to one of these strands of DNA and a reverse primer that attaches to the other strand. You then have the black dot there represents DNA polymerase. DNA polymerase is an enzyme that will use these primers as a starting spot, and they will match up the corresponding DNA thread for the rest of that DNA. And so at the end of the extension step, you have 2 identical strands of DNA because DNA polymerase, again, uses the primer as a starting point to construct a new strand of DNA. If you have more questions about this, let me know. I can explain it in more detail to you in class. I'm going to briefly discuss some of the specific HLA typing procedures that we have, and these are either going to be based on PCR or they're going to be modifications of sequencing. And so one of the modifications that we have for PCR-based typing is called SSP PCR, or sequence-specific primers. Sequence-specific primers are designed so that a product is produced only whenever the primer matches up. That means that you can design your primer to recognize a mutation. And if you have a product, the mutation is there. If you don't have a product, the mutation is not there. And then we visualize the DNA that has been produced in a PCR reaction by loading an electrophoresis gel and applying an electrical current to that. DNA has a net negative charge, and so it will move towards the positive electrode. Again, more information on this in your molecular class, and I can give you more information if you ask me when we're in the classroom. Another modification of PCR is called sequence-specific oligonucleotides. In this case, you denature your DNA and you hybridize it to a labeled probe. One of the differences between a primer and a probe is that a primer is kind of like a start here signal to your DNA polymerase. A probe is also going to match up to single-stranded DNA or RNA, but it's going to have a signal on it. So a labeled probe is a probe that's going to have a signal. A primer for each locus is used, so this happens in separate wells. You have the same DNA, but it goes into separate wells with different sets of primers. And then a DNA probe will allow this hybridized solution to be read and analyzed by a flow cytometer. Basically, whenever this probe attaches to the product based on the primers that were used, that probe runs through a flow cytometer and it is calculated. The third procedure that we use in HLA typing is a sequence-based typing. And so this procedure starts out with a little bit of DNA. We make several copies of the region that we want to look at, and then we run that through a sequencer. And that provides a high-resolution allele level typing. It gives us the exact DNA sequence so that we can look for any polymorphisms or changes. We can tell the difference in the patient's genotype rather than just relying on their phenotype. And this is really important, again, for HLA typing. I'm not going to ask you a whole lot of detailed questions about this in blood bank because most blood bank laboratories don't actually do this sequence-based typing. And the final PCR-based HLA typing procedure is called short tandem repeats, or STRs. So in your DNA, you have certain locations where you have random repeats of a certain number of nucleotides over and over again. So for an example, you may have AG, AG, AG repeated over and over again. And while you may have this AG in one spot on your DNA repeated 14 times, somebody else may have that repeated 26 times. That means that after we do PCR, the length of that fragment of DNA is going to be different. And so we can use this for identification. One of the ways that we use this for identification is in the case of a bone marrow transplant. So we would find these differences. So somebody who has 14 repeats and somebody who has 26 repeats, their length of their PCR product is going to be different because of how many times this is repeated. So what we can do is find the differences pre-transplant, and we can look at these repeats on the donor and look at these repeats on the recipient. After the transplant, we look at what repeats are actually in the recipient. If everything has worked out as expected and the patient has had a full chimerism, meaning that the donor bone marrow has taken over and is now populating the recipient's bone marrow, we should see only the donor's alleles, that 26 repeats after the transplant. If the patient has had a transplant and we see that only 14 repeats are seen, that means that the donor bone marrow did not take over and the recipient's bone marrow that we were trying to replace is still there. And so we use this for information purposes after a bone marrow procedure so that we can look at the percentage engraftment in the chimerism evaluation. Bead chip technology is another molecular-based method that uses short oligonucleotide primers. So these short primers that are attached to colored silica beads that are stuck to a slide. These are amplified and digested DNA binds to these primers. This DNA has a fluorescent label, and so the DNA with this fluorescent label binds to the probe, and then they are analyzed by a computer system, and it will determine which of these colors, which of these colored pieces of DNA have corresponded to each of these probes. And so there are probes available for RHD and for RHCE bead chips. These are not approved for in vitro diagnostic use, but in some cases they can be used for RBC antigen typing. And this final technology is a combination of a qualitative PCR and bead technology. So in this particular case, whenever the scientists are doing the PCR reaction, they use a special C. So remember in PCR we have A's, C's, T's, and G's. This particular technology uses a special C or a special cytosine with a biotin attached to it. The amplified DNA includes this biotin and it's hybridized to probes on color-coded beads, just like in the previous example. But in this case, we use strep-avidin that binds to the biotin in the DNA and it produces a product. This particular technology uses lasers that excite this product and they detect and record the fluorescent signals from this product. This requires a software computer system to analyze the raw data to make these predicted phenotype calls. Again, I'm not going to ask you questions about this. I just want to make you aware of the exciting technology that you may see in the future in your laboratories, especially if you work in a typing laboratory or in a molecular lab. So while in the blood bank, we are basically looking at antigen antisera reactions when we're mixing our something red with our something clear. There are some molecular applications for RBC typing. So these molecular methods in several situations may yield more accurate results and can be more cost effective for donor RBC antigen typing. So while this RBC molecular typing is not really done a lot for our patients, it is done to give us a more accurate picture of the blood that we are giving to our patients from the donors. So in transplantation, we use molecular typing for HLA antigen level typing for transplants. We can also use that for engraftment studies for our hematopoietic stem cell transplant patients. For transfusion, we can use red cell typing in transfused patients. We can determine blood type when a DAT is positive. We can look for some unique situations involving transfusion. For hemolytic disease of the fetus and newborn, we can determine the parental RHD zygosity, so the level of RHD that the parents have, and we can type the fetal blood more easily. We can even type the fetal blood before the fetus has been delivered. For our donor testing, a very important application for molecular testing is looking for viruses in donors that may be below detectable levels by antibody detection methods, which are our older technologies. So we want to make sure that our donor units of blood don't have things like hepatitis C or hepatitis B or Zika virus. We can also use molecular typing for relationship testing, although that's not the primary focus in the blood bank. And that concludes chapter 4 on genetics in the blood bank.